Can everyone hear me in the back? Is this enough volume? Can you hear me in the back? Hey, back. Is that okay? Good, thank you. All right. So good morning, everyone. We're going to continue our lectures on the genome, and today we're going to talk about how our genome is organized and packaged. In the last lecture, I suggested you guys turn off your laptops, and the reason for that, you don't have to do it, okay? But it will help you understand the material better and get better grades. So it's up to you. All right, so we're going to talk about DNA packaging in cells. So the question that we're going to try and answer today is, how is the genome organized and packaged? And the first, we said that human genome is organized into chromosomes, linear chromosomes, and you see here the numbers of chromosomes. We have 22 autosomes and the 2 sex chromosomes. All right, so this is basically a human karyotype. A karyotype for a species is the numbers, size, and the shape of chromosomes, and these are different in between species. Yes? It's not working. Hello, my microphone is not working. I'll just backtrack and tell you guys that the karyotypes are basically the numbers, size, and shape of chromosomes, and the karyotype is unique to every species. All right. So whenever we see a human karyotype like this, what cell cycle stage are these chromosomes in? Does anyone have a guess? What stage of the cell cycles do we visualize chromosomes for karyotype? The chromosomes look like this inside the cell? Usually not. Yeah, go ahead. S phase, when they are being replicated, they need to be opened up and they need to be replicated a lot, and you usually won't be able to see them like this. So what stage of the cell cycle are these chromosomes? Metaphase. These are mitotic chromosomes in metaphase, because that is the cell cycle stage when the chromosomes are most condensed, getting ready for separation, and that's the stage where these karyotype analyses are being done. Okay? All right. So the metaphase chromosomes look like this. So what you have usually in human cells, which are monocentric, that means they have a single centromere. You have 2 sister chromatids that are joined together in the centromere. The chromosomes, these chromatids, which are linear fibers, are completely condensed. Okay, and then they show this X-like structure. So does anyone have an idea why you see this kind of X-like structure in metaphase? How many chromosomes are there in here? 1 or 2? 2 chromosomes, right? Because there are 2 chromatids. Chromosomes are linear DNA, so there are 2 chromosomes here. But they are sister chromatids, so they have arisen from after replication. So what I mean by that is this. What you have in the S phase is you have a single chromosome, right? And it gets duplicated through S phase, and you get 2 sister chromatids. These 2 chromosomes are, during the S phase, held together by a protein complex that has a ring shape that surrounds these chromosomes and holds it together, okay? So this protein complex is called cohesin. It coheses the 2 chromosomes. And then when the chromosomes enter into the metaphase, they start getting condensed, so they start shrinking, right? So they get condensed. And what happens is that all the cohesin from the arms of the chromosomes, they get removed, except the ones in the centromere, okay? Centromere. So what you have is you have 2 sister chromatids kind of separated from each other because they have no longer cohesion, but they're still held in their centromeres because they're going to make the connections to the spindle poles for separation, right? So that what you have is you have kind of like an X-like structure. Make sense? Okay. So that's basically a metaphase chromosome. You have 2 sister chromatids completely condensed and attached at the centromere, and the ends of the linear chromosomes we call the telomeres. Okay? All right. So many years, scientists have been visualizing mitotic chromosomes, and there are some traditional methods that use some dyes to stain the DNA and look at it under the microscope. And these dyes, for example, one example is GEMSA, is a permanent dye that binds to DNA after you subject the chromosomes to heat or proteolysis and yields some bands on it. Okay? And then there are more modern methods called FISH, fluorescence in situ hybridization, and we're going to talk about that next. So this is a GEMSA staining pattern of human chromosomes. So what you can see is these are mitotic chromosomes, and what you see is these bands. So these are GEMSA bands. So a trained person in this technique would be able to tell from the banding patterns which chromosome this chromosome is. Okay, so that's the traditional method of labeling chromosome. And you can see that humans are diploid, so you're going to find 2 numbers of chromosome 14 and 2 numbers of chromosome 10, etc., etc. And if it's a female, there's going to be 2 copies of the X, and then if it's a male, it's a 1 copy of X and 1 copy of Y in normal cases. All right. So the more modern way of visualizing chromosomes is based on fluorescence in situ hybridization. So in this method, it's very similar to a lot of nucleic acid methods. The principle is that you label something, and based on the complementarity of the 2 DNA strands, you can visualize a region of the DNA. Okay, so that's like southern blot and northern blot, all of those things. A lot of them are based on complementarity. So what you're doing here is you want to know which of you prepare metaphase chromosomes, and you want to know which of these chromosomes correspond to a sequence that you know on chromosome 3, for example. Okay, so first what you do is you prepare a probe. So this is a DNA probe that you can prepare by PCR or some other methods. And what you do is you incorporate into the nucleotides a fluorescent molecule. Okay, so then you take this probe, typically a double-stranded DNA, and heat it up and denature. So you release the 2 strands, okay? And then what you do is you also take your chromosomes and heat it up and use some buffers to denature the DNA in the chromosomes as well. So you have these single-stranded DNA, they're opened up, okay? So then you take your probe and you take your metaphase chromosomes and mix it up together. And this probe, it's going to then find and anneal to the sequences that it's complementary to, okay? And then what you're going to see is you're going to see a fluorescent dot at the region where this probe has the sequences are against, okay? So that's how the fluorescence in situ hybridization works. So you will have one probe, you'll have one type of fluorescent dye, and you're going to stain the part of the chromosome that probe is going to anneal. All right, so that's fluorescence in situ hybridization. Any question about this? All right. So this doesn't look like one color, right? There's lots of different colors because all of the different parts of the genome are simultaneously labeled, okay? So this is called spectral karyotyping. That is FISH with combination of colors. So what you have here is instead of using one probe in the FISH, you used many different probes to many different regions in the chromosomes and all different kinds of chromosomes. And what you do is you use combinatorial labeling. What I mean by that is this. So the scientists, there are so many different colors of fluorescent dyes, but there aren't that many. So there are 46 different kinds of dyes that you can label to 46 different chromosomes. So what instead they do is for each chromosome, let's say, they make a different probe here, 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 and here, okay? And then what they do is they use different combinations of the fluorophores. So they can label this with a green and a red. They could do this green and red and yellow, and they could do this green, they could do this red, etc., etc. So by using a combination of colors for each probe, they can tell the computer, okay, whenever you see this combination of colors, label this as chromosome 13, etc., etc. Okay? So that's called spectral karyotyping, and that's the image that you typically see nowadays for human karyotypes, for example. All right. So we said that metaphase chromosomes are easy to visualize because they're completely condensed, and you can see this under a microscope. But chromosomes do exist at other cell cycle stages, but you can't really see them very well because they're kind of decondensed and like just spread out, right? And they're kind of mixed and fit inside a nucleus. So during interphase of the cell cycle stage, interphase is basically the stage that is not mitosis, okay? So during the S phase, during G1 and G2, what you're going to have is you're going to have chromosomes that are fit inside this nucleus. So this is the nucleus of a cell. And all these, they have a highly extended structure, and there's DNA replication and transcription, etc. going on here. In the metaphase, these chromosomes are going to be tightly packed. The nuclear envelope is going to be broken down right before the chromosomes are going to separate. Okay? So how is these chromosomes then packaged inside the nucleus? So the human DNA, if you hold it from end to end for a given cell, is going to be about 2 meters long, and the diameter of a typical human nuclei is about 10 micrometers. Okay, so you want to take this very thin and very long thread, and then squeeze it and package it into the chromosomes in a way that's organized so that when it's necessary to transcribe, replicate, and separate, you can do that correctly. Okay? So think of the chromosomes like a long string, and then you want to actually package that inside a small golf ball, something like that. Okay? So how's that happen? So that happens through a structure that we call chromatin. So chromatin is basically the complex of DNA and proteins that accomplishes the organized compaction of the chromosomes inside the nucleus during interphase. So the first, what you have here is you have your DNA, and the diameter of a DNA is about 2 nanometers long. So the first level of organization comes by taking that DNA and wrapping it around a set of proteins called histones, forming these structures called nucleosomes. And this is what I mean when I say chromatin fiber, because this is basically a protein DNA mixture that is kind of like a fiber, okay? And then this fiber further folds into more compact structures. Some of these structures which we still don't know, it's not very clear. And at the end, in mitosis, you have such a tightly packed chromosome that from the single DNA to the mitotic chromosome, you have about 10,000-fold compaction in human cells. So you really have to squeeze this thing in. Some of these structures were pretty clear that exist because you can actually see. Sometimes in science as well, seeing is believing, right? And then you can use different kind of microscopy techniques, such as electron microscopy, to visualize these things. So this is the DNA under an electron microscopy. You can see it's nice and linear, looks pretty, right? And then the first level of packaging of the DNA into nucleosomes form a beads on a string structure, because you can see that each nucleosome here looks like a bead, and this looks like a little string, right? So you have the beads on a string. And then this chromatin fiber can further fold and make more compact fibers. As you can see here, that is a thicker fiber than here. And then in metaphase chromosomes, you see chromosomes that are highly compacted. So this is the sister chromatid 1, and this is sister chromatid 2. So what you're looking at the surface of a mitotic chromosome, and you can see the chromatin is basically just makes these kind of globule things, and then just and condensed. This is an experiment from 1970s. What the researchers have done is they took mitotic chromosomes, and then what they did is they put that under a microscope with some kind of harsh treatment so that all the DNA was released. So when they looked at this, all of these little gray lines here, it's a little hard to see, are the DNA. And then there was a proteinaceous structure that was left in here, right? And they call this the mitotic scaffold. So the idea is that there are some proteins that mediate the compaction of that DNA into this condensed mitotic chromosomes. Make sense? All right. So I said that there's some question marks here. So we actually don't know exactly how you go from here to here. And this has been a very intense area of research. And that's also one of the things that in my lab I always think about. And there has been a lot of technological improvements, so that allows us to actually look at these structures nowadays. So one of these is called chromo-EMT. So this is electron microscope combined with tomography. So what the researchers done is they found a fluorescent dye that they could take some interface cells. So they take some human cells, put this dye on, and then it stains the DNA. And then they take that DNA and then look under an electron microscope and then see what kind of structures that you see. So these little dark things here is basically chromatin in mitotic chromosomes. So they're very much condensed. And here's the interface nucleus. And what you can see is that the chromatin is less dense, right? Because they are thinner. So these chromatin fibers are thinner. And there's a bunch of other things in the middle here that aren't there in the mitotic chromosomes. What kind of things might be here in this space, in interface cells, in between chromosomes doing stuff? What could it be? Yeah? It could be transcription factors. It could be RNA and DNA polymerases. It could be other proteins that basically read and process the chromosomal DNA. All the things that need to happen inside the nucleus during interface, right? So these are a lot of imaging techniques. So there's lots of nice imaging technologies that are emerging. There are also molecular biology-based techniques that allows you to map chromosomal interactions. So I'm not going to go into detail of these technologies, but here is a highlight from this. What the study is doing is they're mapping all of the genomic regions where two chromatin fibers come in close proximity. So if you know in your genome which two parts have more frequency of coming together, what you can do is you can build a computer model taking those interactions and say what kind of shape are the chromosomes are in based on these two pairwise interactions, okay? So these are the other types of technologies now that enable us to make models of how chromosomes in interface are organized and packaged into the genome. All right, so that's the highlights. That's super exciting, but let's start with the basics, okay? The basic unit of chromatin. The basic unit of chromatin is called nucleosomes. So these are the repeating units of chromatin. That is the basis of these beads on a string chromatin fiber. They are made up of 8 histones that form a core. Here they interact with each other, and the DNA is wrapped around this histone core about 1.5 times. Okay? So there are 2 copies of each histone proteins in here. You have histone H2A, you have H2B, H3, and H4, making an octamer, right? So there are 8 histone copies. Inside our cells, these are the histone proteins, H1, H3, H4, H2A, and H2B. But there's also a 5th histone type that is a little bit different, which is called the linker histone. Okay, so this is linker histone H1. The reason why it's called a linker is that in this chromatin fiber, the 2 beads here, right, so the 2 nucleosomes are combined with DNA in between. And this DNA in between is called the linker DNA. So what the linker histone does is it binds to the linker DNA. In vitro, that means outside the cells, in a tube, if you take a chromatin fiber that is beads on a string and add some linker histone to it, H1 histone, what you will see is that that beads on a string structure will become more compact. So one idea that people have, what the linker histone does, it helps compact the chromatin a little bit more than that beads on a string. Okay. All right. So nucleosomes are not just structural components of the genome. They do this amazingly important job of really packaging and compacting the DNA, but that's not the only thing that they do. They also carry what we call epigenetic information. Okay? They carry information. And the type of information that they carry is different types of post-translational modifications on their tails. What do I mean by that? What I mean is that these core histones, they form that core that the DNA wraps around, but mostly the N-terminal of these histones have long peptides that are unstructured and they stick out of the nucleosome. Okay, so they're like flopping around, and in this picture they're depicted as these dashed lines. The reason why they're dashed line is in the original crystal structures you couldn't see these things because they wouldn't stay at their own place. They would be just like flopping around, so they couldn't in the space know exactly where they are. All right, so what happens to these tails? What happens to these tails is that after histones are deposited into the genome, most of the time, they get modified. Small chemical moieties are added to specific amino acids in the histones. Okay, so this is the N-terminus of histone H3, and you can see that the arginine 2 can have a methylation. Tyrosine 3 could have phosphorylation, lysine 9 could be methylated or acetylated, etc., etc. So all of these type of different modifications, we call them histone modifications, can be added to the tails of the histones that are in the nucleosomes. And these are the type of modifications that are seen very commonly. These are phosphorylation, methylation, acetylation, and ubiquitination. And less commonly, people have found using mass spectrometry that the histones can be crotonylated, hydroxylated, sumoylated, and biotinylated. Okay, so you can add different chemicals to the amino acids in the tails. Any questions about this?
What do they do for like the histone?
Very good question, and that's going to be 2 slides from now. All right. So I said that you can think of these modifications as information, okay, and you can conceptualize it in your head like this. So how is this information, what I mean by information? So there are some proteins that basically take the modification and put it onto the histone tails. So these proteins are called acetylases, methylases, phosphorylases, etc. because an acetylase protein is going to put an acetyl group to the histone tail. Okay? So that's what we call the writers. So that's the writing of the information. Then you can actually erase that information. So there are proteins that actually reverse this reaction. What they do is they remove the modification from the tail. And so these proteins are named deacetylases, demethylases, and phosphatases. They're taking out the modification. And these are what we call erasers. So they are erasing that modification. And then there are proteins that read that information. So these are readers, okay? So what they do is they have a specific protein structure that recognize the modification at that tail. And so these special structures are part of protein domains. So these domains, which some of them are bromodomain, a chromodomain, for example, reads methylated histone tails, a PHD finger, and a W4D repeat, etc. So what do these things actually do? All right, so how do they actually function? There are several ways in which histone modifications can function. So one of the ways in which histone modification functions is that they can change chromatin compaction. So you have nucleosomes here, they're the tails sticking out in this little cartoon. And so when you have a modification, it might actually loosen or compact the chromosomes, okay? So can you give me okay, here's nucleosomes. You have histone proteins. What kind of charge do you think these histone proteins are going to mostly have? Positively or negatively? Positively, because you have the negatively charged DNA, right? And then the histone tails have a lot of lysines. They're also positively charged. But if a protein acetylase comes in and acetylates the lysines, they're going to change that, neutralize that charge. What would your prediction would be in terms of acetylation, making the nucleosomes more compact or more open? More open. So this is not the case for every single acetylation. Okay, so this is not a. So whenever I say, okay, so there are always exceptions to the rules, right? But generally, when you have acetylation, then you have decompaction. And a very good example of that is lysine 16 acetylation of nucleosomes. Because when nucleosomes are compacted, because they are kind of like this hockey puck kind of shape, okay? So you have histones and the DNA wrapped around it. They kind of pack on top of each other. And that lysine 16 acetylation prevents that packing on top of each other because it sticks right out here and it prevents that, okay? So that's one example on how histone modifications can function. The histone modifications can actually inhibit binding of certain proteins. Let's say that you have a certain protein that needs to bind to DNA to do its function. It could be a transcription factor. It could be something else, right? And then if there is a modification in the histone tail, this protein might not be able to bind. So the function of this modification would be to prevent the binding of this transcription factor. You can have the opposite function in which the modification serves as a binding site. It's kind of like a flag. It's an information to this protein, the reader, right? So this is the binder, to say, okay, here I am. You can come and bind and then do your job here, right? So it's kind of an information to recruit proteins. So this is what we call recruitment. So what type of protein is this? Mode binder. Is it a writer or a reader or an eraser? It's a reader, right? And so it's going to read this information and do something. So these type of reader domains are found in proteins that need to go to a specific site in the genome and do some kind of function. So these modifications are telling these proteins, come and bind me and then do your function over here. Okay? So that's how modifications can function. So these type of reader domains are found in various different protein complexes. There are different kind of reader protein domains. There is a diversity of them because you have seen how many different modifications there were on H3, right? There were lysine 9 methylation, lysine 4 methylation, lysine 27 methylation, K4 lysine 4 acetylation, etc., etc. And there are readers for each of these modifications. So these are the type of protein domains that bind to these histone peptides that are shown in red here, and they read it. And some of these domains are amazingly specialized in that they can distinguish a lysine 4 amino acid that has only a single methyl, so that is what we call monomethylation, versus having 3 methyls. That means trimethylation. So these proteins, the way that they interact is so specific that they can actually distinguish if that amino acid is methylated 1 or 3 times. And in fact, and that lysine 4 monomethylation, when it's methylated 1 time, does a different function, and then when it's methylated 3 times, does a different function, right? So you have different information.
Is it the total number of methylations of one tail of one histone, or like the total nucleosome?
So for each nucleosome, you're going to have 2 histone H3s, right? And then they can be methylated in the same place at the same time, or they can have different methylation status, right? They're not always coordinated. Any questions? Okay, so one of the first clues to how these histone modifications work came from the observation that euchromatin and heterochromatin had different kinds of modifications. In human cells, when you stain in the olden days, when you use different kind of stains to stain the nuclei and look at it, the euchromatin appeared kind of white and more decondensed. So euchromatin is decondensed, and it usually has the chromosome DNA regions where there's active transcription and replication, etc. Right? So busy stuff is going on in the euchromatin. The heterochromatin are these dense, dark stained regions. It has much more compact DNA, and they're usually transcriptionally silent, so there's not much going on over here, okay, compared to the euchromatin. And when the researchers looked at the different kinds of histone modifications in euchromatin and heterochromatin, they realized that different modifications tend to associate with different kinds of chromatin, right? So the heterochromatin usually had histone H3 that were trimethylated in lysine 9. So I'm going to refer that as H3, K9, methyl 3, right? Histone H3, lysine 9, methyl 3. Or they had histone H3, K27, methyl 3. In the euchromatin, like I said, you see more acetylation. So acetylation is usually associated with active chromatin. And then you had other type of modifications like phosphorylation. And you can see that methylation can actually be found in both heterochromatin and euchromatin, but the place where the methylation is put. In heterochromatin is K9 and K27, but in euchromatin it's K4, right? So not every time you see a methyl, you're not going to suspect this is heterochromatin. It has to be a specific modification of that methyl in a specific residue, and now you're going to say, okay, this might be heterochromatin. Okay, here's an example for how a heterochromatin histone mark works. So this is H3K9 methyl 3. So what you have is you have a region of the chromosome that you want to condense and form heterochromatin. So the first thing that comes in is a protein complex that contains an enzyme that is a histone H3K9 methyltransferase. So this is a methylase. We call them methylase or methyltransferase. It's the same thing, okay? And it's specific because it only methylates H3 in lysine 9. So what it does is it adds 3 methyl groups to lysine 9, so you have these methylated chromatin, okay? So that methylated chromatin serves as a binding domain for a protein called heterochromatin protein 1, okay? Very imaginatively named. Heterochromatin protein 1, it's called HP1. It has a domain that recognizes this methyl 3 and binds to it. So this HP1 protein also has another protein domain that interacts with each other, okay? So it basically can interact with each other, forming oligomers, so they can oligomerize. They kind of stick to each other. And what that does is you have a chromatin domain where you have HP1-mediated compaction of all the nucleosomes. So these beads on a string, they're kind of got together by the action of this HP1. Does that make sense? All right. So which of these proteins is the writer here? Go ahead. Methyltransferase is the writer. Which of these proteins is the reader? HP1. HP1 is the reader. Which of these proteins is the eraser? We don't see an eraser here, right? So I didn't show that. All right, so here's a question for you guys. If there is a mutation that abolishes the interaction between HP proteins, so that HP proteins can no longer oligomerize, what would the effect be on the heterochromatin compaction? Would it, so if you say that the heterochromatin would be more loose, more open, raise your right hand. If you say that nothing will happen, raise your left hand. Awesome. I'm such a great teacher. You guys all got it. Yes, you won't be able to oligomerize, so you won't have compaction, right? All right. So the heterochromatin can also spread. What I mean by that is when heterochromatin starts forming at a specific site in the chromosome, it can actually spread onto the near regions. And the way that it can do this is based on this property here. All right, so when heterochromatin starts forming, let's say that this is starting to form right here, it's going to be able to spread on this side of the chromatin, and it's not going to be able to spread on this side of the chromatin due to a boundary element. So these boundary elements are bound by proteins that are called boundary proteins, and they are basically refractory to any heterochromatin spreading. So on the other side of the boundary element, you can have active euchromatin that containing acetylation. Okay, so this heterochromatin is going to spread to the right. How does it do that? So when you have methyl 3, you're bound by HP1, and that's interacting with another HP1. But HP1 protein also has another domain that actually interacts with the H3K9 histone methyltransferase itself, right? So it basically has an interaction that recruits the writer of this methyl 3. So when there's a lot of HP1 protein, it attracts this enzyme, which puts more methyl 3. And when you have more methyl 3, you're going to have another HP1 here, right? And then that's going to bring another HMT, the methyltransferase. And that's going to put another methyl 3. And that's going to continue, etc., right? So, and it's going to spread until you run out of HP1 or you encounter another boundary element and forming this nice heterochromatin in between. Make sense? Now, when you go home, think about what would happen if you have different mutations in the HP1 protein, for example, eliminating its ability to recruit the HMT. What would happen? Okay? And this is the type of questions we ask in the midterm as well. So think about that and pretend that what would happen to this process if there was a problem with it. Any question about heterochromatin spreading? All right. Where do we actually see heterochromatin in the genome? I said that there are regions of the genome, they're usually quiet. And so these are the genes that might be, for example, the silenced, genes that should not be expressed in a given tissue, for example. They need to be kept quiet. They can be heterochromatinized and shut off. Okay? And there are other regions of the chromosomes that are specialized and they have special heterochromatin structure. So these structures usually found in centromeres, and then the heterochromatin is also found in telomeres. And they're usually, centromeres and telomeres have micro and mini satellites, right? So what are these satellite sequences? What are they? From my last lecture, I know some of you guys were here. What is a satellite DNA? Is it
when you have like a bunch of repeats?
When you have a bunch of repeats, the same little sequence, 3 base pair, 4 base pair, 10 base pair, repeated over and over and over again, hundreds of copies. Okay, so these are satellite DNA that are also found in telomeres and centromeres. And they serve as the binding sites for proteins that initiate heterochromatin. Okay, so in yeast, for example, the telomeres are heterochromatinized by a protein complex called the SIR complex. So this protein complex has a protein called SIR3 protein, and it binds to the telomeres. How do we know this? We know this by doing experiments using fish and immunofluorescence at the same time. Who has heard of immunofluorescence before? Some of you and some of you not. Okay, so what you're doing here is you're taking your yeast cells and you want to know where the telomere is. And what can you do? You're going to do a fish experiment. So you're going to prepare a probe that has a fluorescence dye, and you're going to hybridize that to the cell where you have the DNA denatured, and it's going to go and bind to the DNA and fluorescence. And you want to actually visualize telomeres. What kind of sequence do you need? What sequence do you need to use to make that probe? What DNA sequence do you need to use to make that probe, to visualize where the telomeres are? Very good. Whatever is complementary to the telomeric sequences, right? So you're going to incorporate a fluorescent dye. You're going to make a probe that is complementary to the telomeric sequences, and you have your cells, and these cells have DNA, right? All kinds of DNA here, and these are in interface, so they're kind of decondensed, etc. You're going to hybridize this probe inside to your cells, and it's going to go and bind to wherever there is a telomere, and it's going to shine, right? And when you look under the microscope, you're actually not going to see all the DNA. You're only going to see the signal of the probe, right? Wherever there is there's telomeres. So that's what this is. So you're looking at the telomeric DNA, okay? And you want to know where the SIRT3 protein is. And the way that you can do this is use immunofluorescence. Immuno, based on antibodies, right? Cheers. He's drinking and saying cheers. So. Oh, good. Good stuff. All right. So we're going to find out where SIRT3 protein is. So what you do is you make an antibody that is going to specifically recognize and bind to the SIRT3 protein and SIRT3 protein only. So you're going to make an antibody that recognizes SIRT3. Then what you're going to do is you're going to take your cells and incubate this antibody with the cells that has all these DNA, and it has lots of different proteins, right? And some of these are SIRT3. And this antibody, when it goes inside the cells, it's going to go and bind to SIRT3. So it's wherever SIRT3, this antibody is going to bind. This is the primary antibody. That is the antibody that's going to go and bind and recognize the target. The target is SIRT3. Then what you can do is you can use a secondary antibody. That these are antibodies that recognize the heavy chain of the primary antibody and binds to it, okay? And these antibodies are conjugated to a fluorescence marker, so a fluorescence molecule. So whenever you have your primary antibody recognize and bind to SIRT3, you're going to see a fluorescent dot under microscope, and so you're going to see these dots wherever you have SIRT3 protein. Sometimes you can skip this use of the secondary antibody, and what you can do is you can conjugate your fluorescence directly to the primary antibody, and you can have a signal wherever your antibody is gone in and hybridized and bound to your target protein. Okay, and so when you look under a fluorescent microscope, what you're going to see is you're going to see those signals, and that's what you are seeing over here. Any questions about immunofluorescence? Okay, so what then you can do is on the computer say, how do these two signals coincide with each other? And when you, for example, look at these signals and make this blue and then this one yellow, etc., and then what you can see is that these little dots appear green, suggesting that the SIRT3 protein is co-localizing. Co-localizing means in the same place as the telomeric DNA. Okay? Make sense? All right. So let's look at a diagram for how the SIR complex actually works, how we think that it works, okay? So what you have is you have these little blue proteins. These are the recruiters. So these are the proteins that bind to the DNA, telomeric DNA, recognize that DNA. And then what they do is they interact and recruit these three proteins called SIR2, SIR3, and SIR4. So these histone tails at the telomeric heterochromatin is hypoacetylated. Hypoacetylation means acetylated less. Okay, very little acetylation at these heterochromatic nucleosomes. Hyperacetylation is lots of acetylation, okay? And hypoacetylation is less acetylation. So you have less acetylation in these chromatin. And what the less acetylation does is it serves as a binding site for these SIR2, SIR3, and SIR4. So these SIR3 and SIR4 proteins can bind to deacetylated histone tails. The SIR2 is an enzyme that is a deacetylase, and what it does is it goes and deacetylates, remove the acetyl groups to the histone tails, then that serves as a binding site to further SIR3 and SIR4, etc., and the heterochromatin spreads. The other property of these proteins is just like HP1, they can interact with each other and form this condensed heterochromatin structure at the telomeres. So which one of these proteins, SIR2, SIR3, or SIR4, is a rider? None of them, right? We're not talking about putting a modification on. Which one of them is an eraser? SIR2, right? Which one of these are readers? SIR3 and SIR4. Some kind of principles. All right. So in this case, which one is the counterpart to HP1? It's SIR2 and SIR4, right? Because those are the readers. The other type of so this heterochromatin in the telomeres is important because it has a function in protecting the telomeres, okay? And it has a function of telling the cells, yes, we have a DNA end that is free at the end of the linear telomeres, but it's not a break, it's normal, okay? So it has a function to protect these ends, yeah?
Would it be SIR3 and SIR4 or SIR2 and SIR4?
Oh, sorry, SIR3 and SIR4, that's right. Thanks for correcting. SIR2 is the deacetylase. So the answer to this question would be SIR4. Okay. All right, so that's the function of the telomeric heterochromatin. And there's another specialized heterochromatin structure that is near the centromeres in human chromosomes. Okay, so they form over the microsatellites near the centromere. So if you look at a human centromere, what you find is you have these microsatellite sequences that are region close to the centromeres, and they're heterochromatinized. And in the middle, you have this region of the chromosome that serves as the centromere. It makes the connection to kinetochore proteins. Kinetochore proteins are the proteins that come and bind to the centromere and also make connections to the spindle so that the cells can, the microtubules can pull the chromosomes and separate. Okay, so that's the function. There's a kind of a very curious thing that occurs at these centromeres. What happens is that these nucleosomes at the centromeres are special. Instead of containing regular histone H3, they contain a slightly different histone that is called a centromeric H3. So they swap this regular H3 with a centromere-specific H3. And that centromere-specific H3 is basically found in nucleosomes that are scattered within this region. And it serves as a site, it serves as a little signal flag to kinetochore proteins to come and bind there. So the centromeres form here. So you can see that there is a histone variant, we call this a variant, It's a little slightly different histone that is specialized for a different function, right? Again, it's signaling to the proteins, come and bind here. All right. So that's the heterochromatin. So inside the interface chromosomes and also in the chromatin, mitotic chromosomes, we see a lot of chromosomal loops. Because imagine this is the chromosome. If you want to make this long chromosome shorter, no matter what you do, at some point you're going to form loops, right? That's the only way you can actually condense a linear chromosome into a small thing. Whether this is packaging in the interface cells or in packaging in preparation for mitosis. So you see these chromosomal loops, and the people have figured out these chromosomal loops exist is basically by doing fish analysis. And what they did is they made probes that are at several. So what they did is they said, okay, this is my chromosome, right? Let's say chromosome 3. And I'm going to take sequences along the linear chromosome and make probes to site A, site B, site C, etc., 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 right? And I'm going to then hybridize each of these probes that are labeled with a different fluorophore and look under the microscope how close they are to each other, right? So if the chromosome is a linear stick, right, if you have two probes that are farther from each other in the linear distance, then in the cell, these should be also farther from each other, right? But if these two sites come together, they're far apart in the linear distance, but close to each other in 3 dimensions, then what you should see is that these two probes, let's say C and H here, would be closer to each other, right, than C and F, which are actually closer in the linear distance, but in the 3-dimensional space, they're farther apart because it's the two ends are forming a loop. Make sense? All right. So what forms these loops? These loops are formed by an evolutionary conserved protein family called SMC complexes. These SMC complexes have their names because they are important for structural maintenance of chromosomes. If you have mutations and problems in these complexes, chromosomes just fall apart. They have a very interesting and unique structure in that you have a protein complex that has 2 subunits. So these are 2 different proteins, SMC2 and SMC4, and a third protein, which we call the bridge protein, also called a cleason, forming a circle. Okay, so this is the cohesin protein complex, the complex that I've been talking about when we were talking about sister chromatid cohesion. What it does, it encircles the 2 sister chromatids and holds them together, right? So the other thing, so these type of circles, these type of interactions we call interchromosomal. Interchromosomal means between 2 chromosomes. These are 2 separate chromatin fibers, right? Each of them is a single linear DNA, 2 sister chromatids. The cohesin can also do this intramolecularly. That means on the same chromosome. So basically holding 2 sites, the end of these 2 loops together, right? So you can have a chromosomal loop, and then the cohesin is holding it together because it can encircle 2 chromatin fibers together. Does that make sense? And all that folding and everything of individual chromosomes, intramolecular interactions are a lot more common, many, many more times more common than intermolecular interactions, okay, in the interface. So each chromosome basically is folding over to itself, right? So in the interface, when you look at the chromosomes and use a fish probe against each chromosome, 1 color per chromosome, and then look where all these chromosomes are, what you see is you're going to have different colors just segregated into different sites in the nucleus, okay? So we call these chromosome territories. Every chromosome has its own territory, okay? It's like kind of like their personal space. So each of them make their own personal space in their chromosome territories. Imagine if you had a bowl of noodles and every single noodle was a different color and they were all segregating into their own little space, you would totally freak out, right? And that's exactly what is happening here. All right, so, and that's due to all these intramolecular interactions mediated by SMC complexes like cohesin. Any questions so far? All right, so go ahead.
With the last slide where there's the creasin versus just DNA binding itself, why are there two different ways that it happens?
Can you clarify it a little bit?
So like that one's binded by the creasin and this one just binds on the spotted one just binds on its own, you said? So like
Oh, yeah. Okay, it's a simplification. It actually needs cohesin. Absolutely.
Okay.
It absolutely needs that cohesin complex protein to make these connections. It was an oversimplification in the textbook. Sometimes we oversimplify to make things clear, right? But good catch. All right, so everybody's on their own territory. So when chromosomes are organized in the nucleus, they're also not randomly just thrown in the nucleus. It's not like your little ramen soup inside goes in a bowl. And actually, certain parts of the chromosomes are going to make connection with the nuclear periphery. Okay, so imagine your certain parts of your noodle always sticking to your bowl. Okay, so how does that look like? It is mediated by a bunch of proteins. So this is your outer nuclear membrane. This is your inner nuclear membrane. So this is the nuclear membrane over here. It has some pores so things can go in and out. And there's some proteins that cover the base of the nuclear membrane. And these proteins are called lamin proteins. Okay, so these are the nuclear lamina, that structure under the nuclear envelope. So those are lamin proteins, and some certain regions of the chromatin make connections with the lamin proteins. There are other type of connections that are made that I'm not going to go through, but basically what you have is certain regions of certain chromosomes, specific regions, are bound by proteins that tether them to the nuclear envelope, okay? And other regions of the chromosomes are not going to have these tethers, so they're going to be inside the nucleus. So this is the how genome is organized in the nucleus, and you can ask, is this important, right? And we know that this is really important because a lot of the mutations of these proteins that do the tethering cause lots of problems. And one of them is really curious. It's a mutation that occurs in the lamin protein. Okay, so this is a mutation. It's a silent mutation that occurs in lamin A protein in humans. It's a de novo mutation, so it occurs new. It's not inherited. So what you're going to have is you're going to have a C to T change at a certain spot in the protein. It's silent. It doesn't change the amino acid, but what it changes the splicing of the protein. What happens is when you have this mutation, this exon 11 gets in the middle of it spliced into exon 12. Okay? And what that does is it removes about 50 amino acids from the protein at the end of this exon 11, right? So you no longer have that because you spliced it out. When you have that 50 amino acid deletion, what happens to the normal protein is that usually this protein gets a modification called farnesyl, addition of a farnesyl group that gets removed. It basically doesn't get removed. So this 50 amino acid deletion here is necessary for the removal of that modification. Because you're missing that due to bad splicing, you basically keep that modification. So what happens? So normally, people with normal DNA and people with normal DNA sounds weird. People with DNA sequence with the normal lamin A sequence, and you have normal splicing, you basically make the protein, and the protein gets this farnesyl group, and that is important for that protein shuttling to the nuclear lamina. And after it gets into the nuclear lamina, this farnesyl group is cut off, so that it can make that nice lamine structure, okay? And you have a normal, firm nuclear envelope structure. But when you have the bad gene, you basically add the farnesyl group and shuttle the protein to the nuclear envelope. But what happens is that when the farnesyl group is not removed, it causes this nuclear envelope to be unstable and kind of dimpled, okay? So if you look at these cells under the microscope, you're going to see this. So this is an immunofluorescence experiment. Lamine A is labeled with a red with an antibody that has a red fluorescence. So wherever you see red fluorescence, you're going to know that okay, there is lamine A here. And what you can see is that it is at the nuclear envelope, right? You see that very nice lamine A surrounding the nucleus. And the heterochromatin associated histone modifications is green. So what they're doing here is that, so you have your histones, let's say H3 here, that is methylated at K9. You have 3-methyl group. You can make antibodies that are going to specifically recognize this modification. And they're not going to bind to H3 that has K9 that's not modified, right? And then you can label these antibodies with green. So whenever you see this green color, you know that there is H3K9 methyl 3 over there. Okay? Make sense? And then when you put the two colors together, you're going to know where the heterochromatin is in relation to the nuclear envelope. And you can see these yellow colors. These are places where specific heterochromatin regions are positioned right next to the nuclear envelope. So these are cells that contain that lamine A mutation that I've been talking about. So the envelope is kind of dimpled and it doesn't look intact. It is not nice and round like this. And the other thing that you can recognize is heterochromatin is inside and it's kind of released, right? So you don't have as much heterochromatin, and the heterochromatin you kind of see that is overlapping, that are kind of tethered to the nuclear envelope, you no longer see this, okay? So that is causing a problem in the heterochromatin formation. And that's also true for other kind of heterochromatin marks, although not as dramatic as this certain heterochromatin mark. And so one of the consequences of this mutation is actually heterochromatin problems in packaging of the DNA. It also causes transcriptional problems. Some certain genes that need to be turned off in these cells, in these mutant cells, they are on, so they're being transcribed. So what's the consequence of this? The consequence of this is a syndrome that is interesting, that's also called aging syndrome. So this is progeria. So these children are born with the mutation, and they age very fast. So they usually die after age 13 to 20, median is about 13, due to problems that actually old people die, like cardiac arrest and stuff like that, okay? And their muscles get degenerate, etc. So it's really curious that a single mutation in the lamina causing so many problems also cause this disease. So there are a lot of doctors now trying to figure out from this how our normal aging might be also a genetic process, right? So it could be due to something that these processes that occur in the syndrome are also occurring in us during normal aging. So they're looking at, for example, nuclear envelope changes during aging, and they do find that in aged people or aged model animals, you see a lot more nuclear envelope problems than in young animals, okay? So that's one way you can use different mutations for learning biology. All right, so that's it for today. There's some links to helpful animations, so go ahead and watch them. They're kind of fun to watch, and it will help you visualize some of these processes and work at home, okay?